Hey biology students, this is Mrs. Miller, and today what I want to review with you guys is some DNA replication. So not only DNA replication, but we're also going to go through the process of transcription and translation. The first thing that's really important to review is what makes up DNA. So what are those three components that we need in order to make DNA, and what does that structure look like? So when we look at the components, one of the main components that we're going to look at are nucleotides. So what are nucleotides? Well, nucleotides are made up of three different parts. So those three parts are the nitrogenous bases. Sometimes we just call them the bases. There's also a sugar and then a phosphate group. So down here we have a nucleotide. You can see our little phosphate group and our sugar. These little guys are going to make up the backbone of the DNA. They're held together with a really strong bond, so they hold together nicely. So you can see they're held together by phosphate to sugar, phosphate, sugar, phosphate, sugar, and so on. So that makes up like the sides of the DNA. The other thing that we're going to see here is going to be a base, and we're going to have a few different bases. So we have adenine, guanine, cytosine, and thymine. So we just abbreviate those as A, G, C, and T. The other thing that's important to note is that this little 5-carbon sugar here is in DNA is going to be deoxyribose. So here's a nice little diagram of it. We have our bases, our sugars, and our phosphates, and that's exactly how they're going to line up. What I like to do is I like to think about DNA as like a ladder. Usually it's twisted up in the double helix shape, but if we untwist it, we end up getting this ladder look with the sides of our ladders that have a very, very strong bond. Here, 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 and so on. And then we also have the little middle bases right here. So the A, the T, the G, and the C. And as you can see, instead of having a nice solid line, they have dotted lines. And the reason why they have dotted lines is because that's a hydrogen bond. And those are weak. It's much more like an attraction, like between magnets, so you can take two magnets and move them apart, so they're not like glued together. So think of DNA like a ladder. The backbone or these sides are going to be made up of the sugar, which here is represented with this orange little pentagon, and then the phosphates, which are the green little circles. So nice, strong backbone, and then the middle is going to be made up of these weaker rungs of our ladder, which are just the bases, and again they're held together with this hydrogen bond, so they are going to be very, very weak because that's just an attraction. As we keep going, we end up getting to Chargaff's rule, and that's just how the bases are going to hold together. So it's pretty easy. A always goes to T. That's why every time here you see a red, it bonds to a blue, blue to a red. And then we also are always going to have G go to C or cytosine go to guanine. So here we have our yellow going to green, green going to yellow. Every time in DNA, that is how they are going to bond together, no matter what, and that's what we call Chargaff's rule. Now what we're going to do is we're going to start talking about DNA replication. So we obviously need to replicate DNA to make new cells. So as you grow, you develop, maybe you get a cut on your arm, and you want to heal that, you need new cells. So we have to be able to replicate our DNA. So the first thing that we're going to do is we are going to uncoil that DNA. So instead of being coiled up like this big chromosome, you can see here the double helix, we're literally going to spread it out and we're going to break it up into its two individual strands. So we're breaking those little hydrogen bonds on the middle. So DNA is split by breaking the hydrogen bonds that hold the bases together. And there's a little enzyme, helicase, that's going to do that. It's almost like taking scissors and cutting up the middle. So we're just breaking those little bonds apart. We know that these hydrogen bonds are really weak, so they're easy to break. So that's why that type of bond is really advantageous in the center. Now, once we have our DNA and it's being broken up by this little helicase, we have an enzyme that you do want to know about, which is called DNA polymerase. To polymerize something means to build something. So DNA polymerase literally builds new DNA. So it builds a new strand of DNA by adding nucleotides one at a time. So here you can see our yellow DNA polymerase. There's going to be one on either side. And what it's going to do is it's going to move down the line on the DNA strand. And it's just going to be boop, boop. And it's going to be popping these little guys, which are the nucleotides, onto the strand. So it's just going to keep going down the line. So as it's moving down the strand, the DNA polymerase also is going to proofread its work and prevent any incorrect bases. So it's always going to make sure that A goes to T, G goes to C. Oops, that's G and G. So then what happens is if it sees any mistakes, it goes back and it corrects that so we don't have any mutations or anything like that. So at the end of this, what we end up with is 2 strands of DNA. One side is original, and one side is new. So like this would be our original strand here on the bottom. This one is our new. So here's our new side. Here's our original. And that's the process of DNA replication. Here we have just a little example about how we use those Chargaff's rules. So again, A always goes to T in DNA and C goes to G. So if I have my original, I should be able to come up with my new side, and I'm just going to connect them with my little hydrogen bonds. So T, its pair is always A, right? C always goes to G. T goes to A, G goes to C. A goes to T, G goes to C, T goes to A, C goes to G, C goes to G. So now I have my complementary strand or my new strand of DNA. So that's our process of DNA replication. The next thing that we want to look at is the differences between DNA and RNA. So we know that DNA right here is going to be double-stranded. So it looks like a double helix. It uses deoxyribose as the sugar, hence the name DNA, deoxyribonucleic acid. And the bases are going to be T, C, A, and G. So A to T, G to C. RNA is going to be different. So DNA is always located in the nucleus, and it tends to be very large because it has the double strand. What happens is with RNA, it's much smaller, so it can move in the cell. It starts out in the nucleus, but eventually it's going to go into the cytoplasm. So first and foremost, RNA consists of long chains of nucleotides just like DNA. So it still has the phosphate group, the sugar, and the base. However, the sugar group is different. In RNA, it's ribose, not deoxyribose. So that makes sense. RNA ribose, DNA deoxyribose. The other big important thing about RNA is the fact that it's a single strand. So where DNA is a double strand here, the double helix, it's only one side of that. And why that's important is because it's small enough now to be able to leave the nucleus. The other big thing is now, instead of thymine, we're going to have uracil. So instead of A to T, it's now A to U, and we're also going to have G to C. One thing that we want to remember is that RNA is considered to be the disposable copy of DNA. So the way that I like to think about it is that DNA is very much like the actual blueprints for, let's say, like a house or something, right? So if you're going to build a house, you want the final blueprints to be held somewhere safe so they don't get ruined. The RNA is the copy that goes to the contractors so they can have it at the house, and if it gets water on it, they spill a little bit of their lunch on it, someone writes on it, it's not that big of a deal because they can always go back and get a new copy of it. So the DNA is the very valuable copy. The RNA is the disposable one. We have 3 types of RNA, and that's mRNA, rRNA, and tRNA. So mRNA, that's the one that we're going to be working with the most. It's called mRNA, which stands for messenger, because it takes a copied message of DNA from the nucleus and it moves it into the cytoplasm. rRNA is a little bit different. So this is called ribosomal RNA. So it makes up the ribosome. So anytime you see a ribosome inside of a cell, it's made up of rRNA. And then the last one is going to be transfer RNA, or tRNA. It literally transfers amino acids to the ribosome based off of a coded message. So oftentimes it looks like this little clover leaf. The one that we work with the most is going to be the mRNA. The rRNA makes up the ribosome, and the tRNA is going to bring in the amino acids. So next we're going to move into the process called transcription. So when we talk about protein synthesis, there's going to be 2 parts, transcription and translation. What happens during transcription is we're going to take our strand of DNA, which again is located in the nucleus. And then what we want to do is we're going to take a copy of one of those sides, but we're going to do it in mRNA. So we need that little mRNA strand because it's smaller and it can leave the nucleus, because this is always occurring inside the nucleus. DNA is too big with the double strand to leave, so we transfer it into mRNA to let it leave. What I like to do is I like to think about this in analogy forms. So think about if you were going to a library. All right, you go to the library and you pick out the biggest cookbook you can. And somewhere in there, you know that there's a recipe for chocolate chip cookies. So you could bring the whole book home, which is heavy and annoying, or you could just take a copy of that single page in the book. So you go over to the copy machine, you make a copy. That copy is almost identical to the copy that's in the book, except it's on a different sheet of paper, right? So this copy of RNA is almost identical to the copy of DNA that you have in here. It's just slightly different because the T's are used, just like the page in the book versus the page that comes out of the copy machine. So in this case, the library is our nucleus. That book is just too big for us to carry. We're lazy. We don't want to do that. So we make a copy of the recipe, which is like our mRNA. And that's the process of transcription. So we go from the big DNA into the smaller RNA. So what I would do here with transcription, here is my side, and this is DNA. The reason that I can tell that we have DNA is because there are T's in there. So thymine is in there. Remember, that's only with DNA. So what we're going to do is, just like when we replicated DNA, we're going to make our little hydrogen bonds to start our new line. And then what I'm going to do is just match these up. So with RNA, remember, A goes to U and G still goes to C. So A goes to U, T is still A, G, C, G, C, G, A, U, G. So now what we're going to see is this little single strand of RNA right here is going to, that's mRNA, now it's going to leave the nucleus. The next thing that we're going to look at is codons and amino acids. So now that we have our little mRNA strand, how are we going to make those into proteins? Well, mRNA has a sequence of nucleotides that will code for a specific amino acid. The code is going to be read 3 letters at a time, and these 3 letters are creatively called a codon. So for every 3 letters, we have a codon. So we have an example right down here where we have UCG, so that would be 1 codon. Our next one you can see is CAC, and then our last one is going to be GGU. So what we have to do is we need to be able to use something called a codon chart, which we see here. And we always start on the left-hand side. And we're going to start with our first letter that we're given here. So I'm going to use this little highlighter. We'll start with our yellow. And what we're going to do, we want to first start with U. So I know that U is right on the top. So I know that I am within this first area. All right, so I'm in the first row. Next, I'm going to look at C. So I find second letter of the codon is C. So I'm in the first row, second box. And now I go G. Third letter is over here. G is the last line. So I drag that over. And I would be right in there. So here. So in that box, all of them are the same. It's serine. I'm lazy and I like to abbreviate, so I just go S-E-R. So for our second one, we have C-A-C. So I'm going to start by going C. All right, I know I'm in this row, C-A. Now I know I'm in this box. And I know I'm the second one over, CAC, so that's histidine. So I'm going to write his. And then my last one, I'll highlight in blue, is GGU. So I'm going to go G, G, so I'm in this area, and GGU is glycine, G-L-Y. So why is that helpful to know about codons? Of course, it's to go through the process of translation, where the mRNA serves as the instructions for amino acids to be joined together to form a protein. The location where this happens is going to be in the cytoplasm where the ribosomes are. So in this case, this little teal blob is going to be a ribosome. And if you remember, that is that's always lowercase, rRNA. And then, so the way that I think about this, now we have our recipe that we've brought home from the library, so our single sheet. Well, what we want to do is now we want to go through and we want to follow our instructions and we want to actually make those chocolate chip cookies. So home is like the ribosome where we can actually cook stuff. Our ingredients are the amino acids that we're going to strain together, and eventually we're going to bake those cookies to make our protein or our final product. So what happens is the mRNA is going to come into the ribosome, and it's going to be read 3 codons at a time. Then we're going to read the next 3, and the next 3, and the next 3, so on and so forth. And what happens is these little tRNAs are going to come in, and they each have one little amino acid associated with them. So they read that code, so CGU, and that brings in a specific amino acid. Then we're going to read the next one, and that's going to bring in a specific amino acid. It's going to go ahead and it's going to build this long chain, and eventually this is going to be the protein. Okay, so here again we have our original transcription translation, what we want to be doing. So in this case, again, we have our DNA, and we want to go through the process of transcription where we go from DNA to mRNA. So A is U, A, C, C, G, A, U, and then we have G. So these little guys all together make up our little mRNA. So this is the copy that we're bringing from the library. So this side is mRNA. Then what happens is we want to go through translation. So what we're going to do is we're going to read these as 3 codons at a time. UAC is going to be our first one. CGA is going to be our next one. And then we have a start of another one where we're going to have 2. We need a third letter, obviously, here. But then what we're going to do is we're going to use that codon chart from the assignment, and you're going to be able to figure out what UAC is. Then you're going to figure out what or CGA is, and so on and so forth. So that's that whole process of going through transcription and translation, and the whole idea of that is to make proteins. If you have any questions, let me know. Otherwise, have a good day.